Hello everyone at SAIAB, hello everyone online. I see that there is a nice audience joining from a few places. I'd just like to welcome you everyone, and especially I'm very happy, grateful, and finally super excited to have Dr. Eleonora Puccinelli presenting to us today at the end of very intense 2-week courses on trophic web ecology and application through stable isotopes. There are a few in the audience that have done the course, so I hope everyone was excited as I was to learn with everyone, and especially with Eleonora and Nokubonga and Lububalo also doing some of the lectures. But today, Eleonora, who is a principal investigator at the Royal Netherlands Institute for Sea Research, is giving us a wrap-up seminar of her research on that she's been doing in the recent few years. I'm not giving the whole history of where Ele was. Ele was here at Rhodes doing her PhD, then she moved to Cape Town, she moved to France, and finally now she is principal investigator at NIOZ. So off to you, Eleonora. Thank you so much.
Thank you, Francesca. Okay, hi everyone. Thank you very much for joining. It's really nice to be back here at SAIAB, back in Grandson, Wakanda. It's like coming back to PhD time, which is bring up lots of nice, good memories. So as Francesca said, I'm a principal investigator at NIOZ. I started last year. And during today's talk, I'm just going to talk about a few of the works that I've been doing before starting NIOZ through various postdoc, and then the work that I've been initiating since last year. But before moving into the actual work, I just want to show you. Wait, here we go. Okay, I just wanted to show where NIOZ is, what NIOZ is. So NIOZ is one of the 9 Royal Institutes of the Netherlands. We have 2 locations, one in Texel, where I work, which is on an island, believe it or not. Seems that the best marine places to work are the farther remote places. And then we have a location in Jessica, which is on the very south part of the Netherlands. And this seems like a big, nice country, but the distance between the 2 locations is 4 hours driving on a very low speed limit. So they're very, very small town. And this is like a photo of NIOZ and Texel. The Netherlands are very well known for being flat countries, if you don't know. Most of this is below sea level. And this is basically the entire part of NIOZ, with the majority of the offices and laboratory facility here. This is Bird's facility, and this is our ports where the fleets usually dock. And basically, NIOZ is very much similar to SAIAB. It's a research institute, so we are not university. And you can get more information about NIOZ if you just Google it, you can find. But basically, just to give an understanding, we are like 4 scientific departments, different disciplines and part of the coastal system departments. We manage long-term monitoring program as very much similar to other national facilities of South Africa. We have a range of laboratory facility in-house, which I found it extremely nice. And the very nice thing that I really appreciate that the national marine facilities sit with us. And we manage the fleet, which are like the 3 vessels that you see here. 2 of them are mostly for local work, whereas the bigger vessel is what we use for more oceanic, non-polar work. In the polar, our fleet doesn't go to. But it's really nice because we can do a range of analyses, which I found it very powerful. And after this, I just want to dive a little bit more in what I actually do. So as probably many of you know, I mostly work on food web ecologies. I started working on food web of coastal intertidal system, focusing on a range of processes that affect the diet of benthic filter feeders, and that focus extensively on these long-chain omega-3 fatty acids, LC omega-3, which is something that is going to reoccur during the course of the presentation, and I will tell you more about it in just a few seconds. Then for the first postdoc at UCT, I started to work on more offshore environment, trying to understand the link between the food produced by phytoplankton at the surface and benthos that sits at depth. And then I move a step further, trying to understand how those food web dynamics may be affected by the nitrogen source used by phytoplankton. Thus introducing more the nutrient cycling, and core pillar of my work has always been stable acid and fatty acid analysis. And when I talk about long chain omega-3 fatty acid, like what do I mean? What are they? They are these compounds are, as I mentioned, so long omega-3 are part of our fatty acid, and they are essential for the correct function of the organism. They play a key role in the formation of the membrane of the cell, and we all need to have them in order to function well. And we know that when organisms have a higher amount of omega-3, usually that is an indication of good food quality. And even if you think about as human, that when you go to the doctor, they say, oh, just get your omega-3 because you're actually going to do well. And similar applies to animal in any kind of aquatic environment or any kind of environment, actually. In the ocean, the main and mostly only producer of this kind of omega-3 and general the main fatty acid is phytoplankton, and consumers, so any kind of consumer from zooplankton to fish to human, cannot produce them in sufficient quantity to satisfy the health requirement, and thus has to be acquired through the diet. And because these fatty acids don't change within the food web generally, and specific phytoplankton groups produce a specific fatty acid, we can actually use them as biomarker and track them in the food chain. And so then they become an extremely powerful tool. So I've applied, so I have three main lines of research, and I want to talk about a little bit, each of them presenting three different case studies, all of them in food web dynamics. The first one is the Benguela project. Second one, looking at pollution effect and coastal system, the Fourth Bay project, which I collaborated extensively with Francesca. And then the last one, we're working on more offshore system, and I want to present the study case of the Dogger Bank, which is part of the recent work I started at NIOZ. But diving into the first one, study case. So the work initiated from, so the general idea behind the Benguela project started from the understanding that if you are in a system that is mostly supported by a new source of nitrogen, such as nitrate, which is commonly available in a upwelling system, this usually leads to a phytoplankton community characterized by big cell, such as diatom, which are very well known to produce a very high amount of lipids and of omega-3. And thus we can expect to have high transfer to higher trophic level. Whereas if we are in a system that is mostly supported by a recycled source of nitrogen, such as ammonia, this may lead to a phytoplankton community characterized by small cell, as a cyanobacteria, which are known to produce very little lipids in general, or fatty acids in general. And thus there will be less transfer to a higher trophic level. So we hypothesized different kind of scenario of omega-3 production as a function of the nitrogen source, led by nitrate, led by ammonium, or a mixed scenario, which then the smaller trophic community will play a big role. And these were summarized in the review paper that was published a couple of years ago. And this set the ground for the Benguela project. So the Benguela project, which of course was developed in the Benguela upwelling systems on the west coast of South Africa, aimed to look at the omega-3 production and transfer within the system. So we wanted to see which one was the main nitrogen source used by phytoplankton, what was the amount of omega-3 produced by phytoplankton, and how much is transferred to zooplankton. And so to do that, we conducted an investigation at the anchor station in Santa Helena Bay. specifically in Elands Bay, where we sampled the same station for 10 consecutive days in the hope that we could sample an upwelling cycle. This was done during the upwelling season, which was March 2020, just before COVID, literally, before COVID kicked in. And we collected a range of samples from a physical to biological analysis. We ran a range of experiments for the carbon and nitrogen uptake rates, as well as fatty acids production rate, and then there was a laboratory component. And we were lucky enough to sample an upwelling cycle on day 5, 6, and 7 of our investigation, which is really, really nice. We didn't know, and we managed to sample it. But let's dive into some of the results. So here I'm going to present the same kind of plots for this session of the presentation. You always find depth on the y-axis. That was about 30 meters at the anchor station, and time on the x-axis, so from 1 to 10 days. Each of these dots correspond to a sampling station, so we sampled every 5 meters every day. And as you can see, in general, that so for the chlorophyll concentrations, for instance, chlorophyll was extremely high before and after upwelling. So the upwelling is indicated by the black box. And the water column was quite well stratified, approximately, like we could see a thermocline between 5 and 7 meters. And was very low chlorophyll concentrations at depth and during upwelling. Similar pattern we observed with temperature. Temperature is cooler during the upwelling event, which is expected when you have this upwelled water coming from the bottom, and also potential density. And this pattern was present across all the physical parameters measured. When we actually got to look at the nitrogen source used by phytoplankton, we found again a similar pattern. So here I'm presenting nutrient concentrations for nitrate and ammonium, so the two main sources. And I want to stress that the scale are very much different between the two. And in general, we see that nitrate is more abundant than ammonium, and but both of them are upwelled to the surface during the upwelling events, whereas nitrate is extremely abundant at depth, with up to 30 micromolar, which is extremely high value for nitrogen coastal system. And when we move to look at the actual uptake of nitrate and ammonium, so how much, so for uptake, I mean how much nitrate or ammonia the phytoplankton has uptake in the body to basically make photosynthesis and produce organic matter. We can see that nitrate is again, the uptake for nitrate is much higher for ammonium, scale is different again. And while ammonium is quickly utilized once it's available because that is energetically more favorable, that was quickly uptake. There was not much in the water, and actually nitrate was the main source used by the phytoplankton during the investigation. And when we move to look at the amount of lipids, amount of fatty acids present in the phytoplankton that we sampled, again, we found a similar pattern as for the previous parameter. Amount of total fatty acid and omega-3 were extremely high at the surface and subsurface before and after upwelling events, up to 200 microgram per liter of total fatty acid, 30–40% of which were omega-3, which is an extremely high value for any kind of system. And then the nice thing that to me is really strike me is that the amount of lipids was extremely high before upwelling and after upwelling, and there was this gap during the upwelling event, but the system recovered within just 2 days post-upwelling. And we know that phytoplankton is favored post-upwelling, but the speed at which this process actually occurring is extremely remarkable. Even more remarkable in my way is that before upwelling, there was a very high amount of non-diatom trophic marker like dinoflagellate or aptophyte, and there was a switch to diatrophic marker post-upwelling. And again, It is known that diatom are usually favored post-upwelling because a nitrate, iron, and other range of other nutrients are upwelled to the surface. But again, at the speed at which and the way that this actually process is occurring within just a couple of days, it's super nice to see. So this work has been published last year. The zooplankton part is still on the go. It's not as straightforward as the phytoplankton, so there is a bit more thinking to do. But this set up the ground for way more further exploitation or exploration of upwelling system, especially on lipids, because to me, they're very much understudied regions of omega-3 production. So this was the first line of research, and I just want to quickly move to a different one, which is pollution inputs into coastal area. And this is part of the False Braids project, which was very much a multidisciplinary, multi-institutional project that I led while I was at UCT. But in general, when we talk about coastal system, they are, as you all know, they are extremely important for a range of reasons. They provide food resources, protection, nutrient cycling for water quality. However, these systems are subject to a range of threats, including pollution input, increasing level of urbanization of climate events. And this basically, within this framework, we basically developed the False Braids project. That we basically look at the role that filter feeders, specifically mussels, may have in mitigating pollution effect into an urbanized system. And we use False Bay as a study case. In the specific, we decided to choose False Bay because False Bay is is the largest Southern African natural bay. It is extremely important from a socioeconomic perspective. There is a range of tourism. There are a range of small-scale fisheries occurring within the bay. There are a range of pollution sources within the bay, from river outflow, wastewater treatment, sewage. So it's nonetheless subject to a high level of anthropogenic activities, as well as is subject to, there are over 2 million people living in the bay, so the level of urbanization is particularly high. And at the same time, the coastline is dominated, the intertidal, especially intertidal coastline, is dominated by the presence of the mussel Mytilus provincialis, and so that represented the best, basically, avenue and the best place where to actually look at pollution effect in coastal system mitigations effect as well. And so with the projects, or the first part at least of the project, aim to provide baseline information on pollution input into the bay, evaluate potential anthropogenic effects on the natural environment, and characterize the role of mussel in attenuating such pollution effect. So the sampling was conducted over 4 times. So this was done in 2018, but the sampling was repeated 4 times within 2018 and 19, and we collect a sample from 8 rocky shore sites, distributed, like openly distributed within the bay through offshore system. So we look at transit moving away from the rocky shore sites. We had also an aerosol component, and then, of course, there was a laboratory component with all this range of analysis were conducted afterwards. And I want to provide a few of the key results, because then highlight a little bit more of what we actually did within the project. So here I'm reporting the nutrient concentrations within the bay. The different color correspond to the 3 main nutrient sources, so nitrogen, ammonium, and urea. Urea was included in this case because this was a coastal system in which we expect to have urea. In fact, we did. That also plays quite an important role within the nutrient cycling. But generally, what you can see, so the sites are numbered from 1 to 8, and basically it's from west to east. And in generally, the majority of the nutrients were extremely high at the site 5 and 6, which you can see in the map in the corner. Those correspond to the area within the Gordons Bay area. And when we look at the stabilizer composition of the nutrient of the PON, so particularly organic and nitrogen, and the one of the mussels. And this was done to investigate potential pollution effect related to nitrogen. In this case, what we observe is that nitrate, let me see if I can get this right. So nitrate, the nitrogen composition are indicated in blue as a nitrogen composition, let's say, of approximately 8. The PON had a nitrogen composition of between 6 and 8. However, if the phytoplankton were only using nitrate as a marine source, we should have expected the PON to have a nitrogen composition of approximately between 2 and 4. But this did not happen. Implied that there was a nitrogen source entering into the pool, and the nitrogen source is most likely anthropogenic, a waste or other nitrogen source from fertilizers, so from wastewater that have a way higher nitrogen composition as a source. So this is an indication that specifically at these two sites where we observe very high nutrient concentration, we had pollution input from a nitrogen source, most likely fertilizer, agricultural fertilizer. Then we look at pollution input in terms of metals. Here I'm reporting data for mussels and sediment from intertidal and offshore station. Mussels are in yellow, the sediment is in blue, that blue, light blue onshore, offshore. And we are reporting a few selected metals, and the red line indicates the maximum permissible level for shellfish consumption. So level for that specific metals. And as you can see, I don't have it for all the metals because as Rakubanga, we discussed this quite a lot. There are no available guidelines for a lot of this metal. So in this case, what we observed that we had a very high concentration of specific metal at site 5 and 6, which is again the one that are located in the Gordon Bay area. Yeah. I'll put it, especially for zinc, mercury, lead, arsenic. Arsenic was also very high in a range of other samples, and all of those were generally in the mussels. So again, there was another pollution input source, specifically in the Gordon's Bay side. When we actually go to look at the how the animal actually doing in this condition, then the situation slightly changed. So this is a result for the biodiversity associated to mussel bed, and we did observe variability amongst the different sites, but there was no significant effect. So the sites did not differ from each other. Site 5 did not have mussel. It could have been that the particular rock shore is quite a flat rock shore, perhaps is not the best suitable environment for mussel to settle, or perhaps there is another pollution effect, we don't know, but that's basically the resultant effect. And when we look at the condition index of the mussel, which provide basically a proxy for the health of the mussel itself, we actually start to observe quite a clear cut between the west and the east. So animal in the east had a much higher condition index than animal on the west. And when we see the fatty acids composition of the animal, which again provide an information on the health of the animal themselves, again the plots they separate between east and west, with animal in the east side having a high concentration, a high amount of a range of fatty acids, which you can see here, which are typical of diatom and unflagellate trophic marker, which are indication of a good food source. So it seems like the animal on the east were actually doing much better than on the west, but there was no connection to pollution inputs. So this was just a snapshot of the kind of analysis that we did. There was also pollution input from aerosol, and this was summarized in the paper that was published a couple of years ago. But basically what we observed, that anthropogenic input was indeed present on one side of the bay, but such effects were not really reflected on the mussel, both adult or early life stages, which I did not present, but the pattern is the same. But the coastal circulation, I think that it plays an extremely important role in False Bay. False Bay is a fast-flowing bay that have an anti-clockwise water circulation. But a lot of the nutrient actually gets retained within the garden base area, and this is probably why we kind of observe the majority of the pollution input in that specific side of the coast. This was one sampling event. We have other three, but our recommendation is that there should be a long-term monitoring program in place, which I believe either it's already or is about to be put into place. But this will provide a better information, especially for such an important socio-economical hotspot for South Africa. And the last part that I want to talk about, okay, so I'm good with timing, is the work that I've been doing since I'm at NIOZ, and especially in the North Sea, on a specific area, which is the Dogger Bank. So the North Sea, it's a very different environment from South Africa. Is the sea located in the part of the northern part of the Atlantic? It's very shallow sea. I think the maximum depth is like between 80 meters and 100, so it's very, very shallow. But at the same time, it has quite a complex food web that is influenced by a range of top-down and bottom-up processes. And if we go to look a bit deeper, we'll see even a shallower feature, which is this Dogger Bank, which is a feature in the middle of the North Sea with approximately 20 meters deep. But because of the shallow depth, this actually area, it's a very high hotspot of biodiversity and productivity for the region. A range of animals actually whose colony is on land actually feeds in the Dogger Bank because of this high productivity from plankton up to fish. And so it's an important hotspot within the region. However, the Dogger Bank is subject, the North Sea is subject to a range of human threats, including overfishing, large development of wind farm, oil and gas extraction, pollution, and of course, global warming, which is at such a shallow sea, you see the effect quite strongly. The North Sea is quite important for a range of apex and top predator animals, which I wasn't very used to work until I got to NIOZ, but I collaborate with people for this part. And you can clearly see that the concept of central place foraging. So a lot of the animals, so the animal that feeds in the North Sea, they know exactly where to feed. And the place where they're going to feed is tightly linked to where the colony is. So here I'm presenting like a seal of gray seal, harbor seals, or tracking data or gulls tracking data. And as you can see, they have a specific route where they go to the Dogger Bank and back, so they know where to go. And most importantly, especially if you're looking for the harbor seal or even the gulls, the distribution, so the colony are very spatially segregated from each other. So animals don't really overlap. So animals from different colonies don't tend to overlap. And what becomes very clear is that if for any reason the food available for this animal may change in the near future, they will be forced to go somewhere else or to feed on something else. And this becomes extremely costly for this animal that already need to make a journey to feed every day or every few days up and down from the colony down to sea. So also the Dogger Bank itself, especially within the last, or the North Sea in general, within the last 40 years has been subject to quite a clear increase in seawater temperatures, but productivity as well as fish recruitment or small copepods of a range of parameters have actually decreasing over time. When we actually go to look at the data available for the North Sea, and now you were saying it's in Europe, surely is very well studied, but actually it's not as well studied as I thought, at least. So some areas better studied than others. There is limited temporal variability in the data collected, so it's a lot of those are snapshot. Very often there is a lack of depth resolution. It's only one trophic group or one species even studied at the same time. And this creates this connection of data, which is very difficult to integrate, especially when you try to understand the system. And so within this framework, this basically set the ground for the ACNAU cruise, part of the ACNAU project. ACNAU is a EU Horizon project, which is coordinated by NIOZ, by my institute, where they're looking at biodiversity threat indices, and assessment, and within the European regions, both from the North Sea, Baltic Sea, and so on, but also Mediterranean coastal water, and actually the Black Sea as well. But within those ACNAU projects, the ACNAU cruise was developed in the North Sea to basically have a better understanding of these integrations of across different trophic level specifically within the Dogger Bank. And we use very much an holistic research approach. So the cruise was conducted in June 2023 in the Dogger Bank, and here I'm presenting like the cruise trajectory. We have 8 transit. Each of these dots correspond to the different station and sample, and the transit were set to target megafauna foraging hotspot. And so basically we look at seal tracking data, which are indicated by all these agglomeration of red small dots, to target a specific area where we knew we should have found apex in the region. And so during the cruise, we collected a suite of variables looking at the phytoplankton or bacterial community composition. Zooplankton was collected with a net and with a pisco camera. We had permission from benthos and demersal fish using a troll. EDNA samples were collected for both pelagic and demersal fish identification. And then, of course, mammal seabirds were collected during the cruise by visual observation. And I want just to present a few of the results that just define a picture of what we observed during the cruise. So here are the productivity data presenting in the first part, chlorophyll concentration, used as a proxy for productivity. The graph indicates the depth, with chlorophyll concentration, and the color coded by station location, so from north to south. And in general, we can see that chlorophyll was generally low throughout the investigation. There were variability between north and south, and most of all, there was variability at time. Chlorophyll was increasing with depth, which is expected, especially around the depth of Fmax, of maximum fluorescence, but generally concentrations were low. When we look at the amounts of phytoplankton present in the water, we actually start to see some interesting pattern that stations that were located on the banks, on the shallow parts, had a higher amount of cell in comparison to the offshore system. So this indicated that most likely we were observing 2 different communities of phytoplankton between the 2 regions. And you need to trust me here because it's confirmed by other analysis, but usually the different phytoplankton community that we observe may probably lead to different food quality available for the higher trophic level. When we focus on the zooplankton, in this case, we are just going to present you data for one bank station and one offshore station. Looking at the offshore stations here, in the different panel, top panel is indicating temperatures, and we can see that the water column is very well stratified, very high temperatures at the surface, very low at the bottom. We have an Fmax, so a depth, a chlorophyll maximum at depth. We don't observe any diatom change within the region, but all the copepods are clearly sitting where the Fmax is. And this was valid for fish larvae, for a range of other parameters that were observed. And in this photo gray here, and it looks a bit odd, but each of these dots correspond to a copepod. This is what is observed in the camera system when we had on board. Whereas when we actually look at the bank station, we start to see that water column is not as well stratified. Definitely there is no Fmax. Chlorophyll is very nicely distributed, concentration distributed throughout the water column. There were a lot of diatom chain, especially close to the bank, and there were no copepods involved. And here in this photo here, all these stringy parts are represented by the diatom chain. So again, basically what they are observing that there were very much 2 different communities between the bank and the offshore system. When we move to fish data, so the target fish species was the sand eels because it's one of the prime food source for a range of birds. So here you can see puffin with all this fish in his beak. All of those are sand eels. Sand eels, they are migratory fish. They sit in the water column during the day and they bury themselves in sediment at night. And that's how we collect them. We collect them at night with a trawler. And you can see that they were extremely abundant along the slope of the bank throughout the investigation. When we focus on some of the megafauna, here I'm representing, I took 3 species, but this applies to many others. So Haboporpoise, great seal, and puffin. So Haboporpoise, the belly are present. We only found them here. Puffin was not observed throughout the investigation. And this is the main ground where you find puffin all the time. And great seals were present, but not in very high abundances, as in previous year. And in fact, when we compare to previous year's data, which we have for some species and not for others, we can actually see that gray seals, but all of the other species were extremely abundant, especially if we think about Haboporpoise and puffin. So what happened in 2023 that was so weird that we saw such a pattern where productivity was extremely low, water column was very well stratified, some species were not present at all. We actually had at sea during the most intense marine heat waves that was observed in the North Atlantic, which happened to be June 2023. And you can see here with a big peak of temperatures of sea surface anomaly. When we actually go to look at our data, so here is a short video that's the first part, indicate the sea surface temperatures for the month of June. As you can see, they keep increasing. This is the cruise tracking positions where we sample. And again, when we look at the depth profile, we can see that the sea surface temperature was extremely high with a thermocline of about 10 degrees, especially in the offshore station. Thermocline of 10 degrees is huge. This is an extremely high thermocline, especially if you look at the depth of this area, which is only within 20 meters. And so it is known that marine heat waves are more and more frequent, but what we start to wonder is this usual condition, or is just actually becoming the future norm? So is this what we're going to expect on a daily basis? So we probably were not able to observe animal because this was not the right environment for them to thrive. And so they just avoided the area and they went probably somewhere else. So just in summary, we sampled during the most intense heat wave recorded in the North Sea. There was low productivity, different phytoplankton community between the bank and the shore area, sand eel most present on the slope, and unusual megafauna distributions. This is very much a work in progress. Some analysis, most of the analysis actually still needs to be done. But what I want to be the take-home message from this work is that we use this holistic approach to have a better understanding of an ecosystem across different trophic levels. It is costly. It is logistically extremely challenging to organize, but it's the only way forward if we really want to understand how an ecosystem works. We need to account for temporal variability, which we of course did not do in one cruise, and we must integrate with tools that we already have, like tagging, satellite, or time series, because this is going to be the only way to get reliable data that can be fed to policymakers to have a better management and policy, in this case for the North Sea, but this applies to any other kind of marine system. So, and with this, I just want to thank you all for the attention. This is not a one-person job at all. I have worked with a range of beautiful people in my career, and I'm very grateful for that. I like 3 of them because those 3, they really helped me a lot over the last few years. And I think if I got this position at NIOZ, it's also thanks to these 3 people. I also want to thank specifically Francesca Nenokubonga because as you all know, I was here for the course, the stabilizer best practice course on food web studies, which was funded by SCORE, but as well by SAIAB. And it was really a fun journey, and I'm very, very happy to be back here to have worked with you on this. Thank you very much.
Thank you. Thanks, Eleonora. I don't know if I'll take some, we usually start with the floor. I'll take some questions from online, actually, if there are. Can I stop sharing? Yeah, so that we can see. There are no questions. Or maybe there's somebody. I'm surprised. Okay. All right. Then we move on to the floor. Any question for Eleonora? Yes, and I think you might have to come here to just so that. Oh, should I repeat the question? Or for the yeah, either ways, or for the volume, for the people online. Yeah.
Thank you very much. Yeah, that's great. I'm more curious about the pollution in False Bay.
Yeah.
What's the main source of the arsenic? And then what was the reason why it was so high at Simonstown when there was no other real pollution on that side?
So for the arsenic, so Simon's. Okay, sorry. No, Simonstown, we were sampling close to the marina.
Okay.
And I think the marina, which is the same actually for Gordon's Bay.
Yeah.
So I think a lot of the inputs come from, especially for metal, comes from that.
Okay.
And also for a lot of so some of the metal are still used in antifouling painting, which is banned according to law that was developed in 2014. But a lot of the boats still have the old antifouling painting. And so I think there is still an effect on that.
Okay.
Yeah.
Cool. Thanks.
Yeah, sure.
Thanks, Ele. Any more questions from the floor? I do have a couple, but basically with your last slide, you touched on them because I was seeing, especially for case 3, all of that efforts of getting, in terms of funding as well, getting that cruise together and you advocating for a long-term monitoring program that takes that considers all of those aspects. Is this an effort that is run from your institute, or how does it work? I know that you said it was a Horizon program, but is it the core of the project directed and managed by your institute, and how does link to that your institute link to that big problem that you showed us that is happening on the North Sea? It's looks like quite a messy multifactorial type of problem.
Yeah. So that part, specifically the Dogger Bank, a little fraction belongs. So the North Sea is divided. There is no international water. So part is UK, part is Dutch, part is Germany, part is Norway, so everyone got a piece. And the majority of the Dogger Bank actually is managed by the UK. So it's all UK water. So within the project, we have UK, so there are UK, Norwegian, so there are partners from every institute, which especially the UK were on board. And they have more cruises, especially the people from the UK on monitoring fish. So that's why they're quite big on that. But from the Dutch side, there is quite a big push from the government because so the Dogger Bank is an MPA, but there are wind farms in the MPA. They're going to be developing more wind farms in the MPA. So there is quite a strong push to try to understand the effect of the wind farm from the Dutch perspective. And so this was a EU-funded project, but we have other programs run by the government, the Dutch government, that push towards more research in the sea. So that's the thing. There is still, in my view, not enough coordination among the different countries, and this is a problem that we all raise. And this new project was trying actually to fill a bit those gaps. Yeah.
Thank you very much. Gigantic
effort,
effort.
but It's not my project. I'm part of it.
Yes, there's one here, Camille Kubonga, please.
Yeah, I have two. So you said, if I'm not mistaken, I know very little about upwelling.
Yeah.
So you said during upwelling, there's nutrients coming to the surface. So what does that mean to the deeper ocean species? Would that have an influence in terms of their feeding behavior?
So, like for deep ocean species, how deep are you talking about?
I don't even know the deep sea.
Okay, so for deep sea, we refer to well, in a very simplistic way, it's anything above 200 meters of water. So in this case, so I haven't now worked on deep sea species for the upwelling part. I did do a work on the Cape Canyon, which is a canyon which is just off the west coast. We actually look, which is influenced by upwelling. So canyon is a bit different features because you have other issue to account for, not only for upwelling, but I think there is an effect of upwelling on the food available to them. But for that particular project, we couldn't separate. There were two confounding factors.
Okay.
So I would expect that perhaps yes, but I don't know. I think it's very much system dependent because there are more factors that plays a role.
So also with the species that feed on the Donga Bank?
Yeah.
Donga Bank, yes. It's all the anthropogenic influence and climate change.
Yeah.
Has there been any observation on their movement in terms of movement patterns and also feeding behavior?
So that there is an entire team that works on this seal, especially seal tracking data or birds tracking data for looking at that. I'm not entirely sure if we can clearly say that. I think they have also other issues. At times the population vary a lot in terms of abundance. Sometimes they're extremely fat, some years they're extremely slim. So I think what is actually affecting is the food available for them. They can't move, or they don't really, their colony is dead. And so I think what we're going to see is that the food available for the species actually is going to decrease, or the quality is going to decrease. Because actually what happened last year, we went to sea in June, and then in September we start to see quite a high mortality of certain species of birds along Texel, where we live. Texel, it's we have a lot of birds. It's one of the UNESCO sites. There are a lot of migratory birds coming in and out, so it's quite an hot spot for bird ecology. And chatting with colleagues about it, it is common to see mortality of birds along our coast, but not so early, not just after the summer. And so what they were thinking is that those animals were actually feeding on something that was not energetic enough for them. Either the fish was not fat enough, or there was some species were not abundant. But places, so for the big, so it becomes a bit tricky. But there is they do monitoring. Yeah.
Thank you. Let me see if there's anything from the chat. It doesn't look like. And there's one more. Yes, I thought that people are getting less shy. Yes. The new one.
Yeah, I have a question here. I'm more freshwater, so little things and so.
That's fine.
My question pertains to you mentioned the use of like the third case study, construction of offshore wind farms. As a lot of governments are pushing towards green energy and all these things, how much I know it's still a work in progress, like you mentioned, how much damage do these offshore wind farms actually doing? Is it really a sustainable source of energy, or are we just moving from one problem to another problem?
I don't know. So they're trying, when they implement the wind farm, they always try to make them in a way that they can be used for something else. For instance, aquaculture of bivalve works really well in the North Sea. Is not as rough as the ocean, but they do aquacultures for bivalve, oyster, so mussel, oyster, or seaweed aquaculture. So they're trying to compensate a bit on that. But the point that I think at this stage, there are a few studies that are looking at the effect of the wind farm from by the noise, by looking the shade that they create in the environment, especially when you have the floating one. But I don't know. I think it's it's something that you need to balance out, accounting for all the partner that all the aspects, like how much a wind farm lasts, how do you replace it, how do you what happened to the wind farm once you need to replace it. So I think there are a lot of factors needs to be accounted for, but at the moment I think whoever's done that kind of the numbers seems it's better than actually fossil fuel. Fossil fuel is really a big thing, and especially there was the first time that I've gone into the North Sea, and you literally go and see this massive oil and gas extraction, and it just there are so many that feels like you are in a movie. That's what's my impression. That I never really one of these old like 10 of them on your eyes, and it's
it's like choose your least problem or the least damaging problem.
Yeah, there are a bunch of projects, especially with economies, that can balance out from a CO2 perspective or greenhouse gases that can kind of balance out benefits and cons. So I wouldn't know, honestly.
Thanks so much. If there aren't any more questions, I would like to go for another round of applause for Eleonora, and thank you for your Bravissima.
Le due parti, la cosa.
Ah no, voi